We're live.
Hey, what's up, everybody? All right, so I'm going to be stepping in as guest moderator tonight because as the 2022 debate challenge continues, Donnie just couldn't help himself. He's got to get back in the ring. So tonight we're going to be debating endogenous retroviruses, and I'm hoping that everybody here knows what those are. If you don't, these ERVs are just DNA sequences that are present in germlines of non-viral organisms. That's important. So they either resemble a virus or can come from a virus, and they're estimated to be anywhere around 8% of the genomes. The topic is going to have big implications for the ongoing creation evolution debate. So with that out of the way, the debate tonight is whether or not these DNA elements are enough to support the theory of evolution, the idea that common descent is true. So he's going to have a little bit of bird proof tonight. He's going to take the positive. He's going to try to definitively show that there that is able to explain these ERVs to the same degree that common descent can. So in this case, Donnie's going to have to show that ERVs either do not support common descent at all, or that there is another theory, namely creation, that can plausibly account for the existence of these ERVs. So get ready. This is going to be a good debate. So pretty technical. These guys are no rookies. He's had almost 100 debates. Snake's no stranger. I know a lot of you guys out there know him. So let's give the debaters some grace in the chat. We don't need to be attacking these men personally. There's no need for that, as the mod will be, ridding the chat of any irrelevant babble. So as for the format, we're going to do 12-minute openings for both guys. They're going to have 8-minute rebuttals. They're going to do around a 30-minute discussion specific to ERV. 5-minute closings, and whatever time is left, we'll try to wrap it up around 2 hours. But whatever time is left, we'll save for Q&A. So with that, I hear Taylor's going to be going first. Is that right, guys?
All right. Maybe we will. We can do a couple intros before the opening statements if you want. Snake, you can kind of give the audience a little rundown on who you are and your channel. Brandon, I think it might have improved, but you were kind of glitching there a little bit for about a minute. But it looks like it's improved. Or was that just on my end, Snake? Was it glitching for you a little bit too?
Yeah, for a second, but it wasn't too bad.
Okay. So I think we're good. And yeah, Snake, if you wanted to give a brief intro, who are you, a little bit about your channel, so on and so forth.
Yeah, so I am a molecular cellular developmental biology student, and my channel is Snake Goes Right, and it's basically about various topics, but the central theme is kind of, well, the name is derived from the biblical serpent, but the theme shares a common ancestry, if you will, with that same story, and which is basically that knowledge is worth the cost, or knowledge is always the best way forward. So, and knowledge of good and evil is a good thing, I think. So, and that kind of applies to a lot of things that I'm thinking about, not just scientific, but moral and political. And I do take inconvenient political stances that don't necessarily align with the stereotype of an atheist, things like that and such. And yeah, but I have a strong background in science, and that's what we're going to tackle tonight.
Okay, much appreciated there, Taylor. For me, I'm Donnie, as everybody knows, or you may know me as Standing for Truth. Although I started this ministry several years ago, we now have a team Standing for Truth. So I'm excited for this. I believe this is formal debate number 98 for myself. I know Snake has had a ton of debates as well. And I specifically like debating these types of topics. The more challenging topics. I've had plenty of debates on ERVs, chromosome 2 fusion, Neanderthal phylogenetics, those types of topics that deal with arguments from the evolutionist side. that they would say essentially preclude biblical ancestry or young earth creation. So I like tackling the tough topics, and tonight is endogenous retroviruses. So I very much look forward to this. So that's my intro, and I'll hand it over to Brandon, and I guess we can kind of get into some openings.
Excellent intro. So like I said, we'll start with 12-minute openings, and whenever you're ready, Taylor, I'll go ahead and start the clock for you.
Yeah, all right. So yeah, we had a brief explanation of ERVs. They are identifiable by certain viral genes that are part of them, and they insert into the genome. That's how they reproduce. Sometimes they'll insert into the gametes or reproductive cells, and thus everything in the surviving lineage of those reproductive cells will have these ERVs. So you can essentially trace who's related from that. And the basic idea is that we share so many retroviruses in the exact same locations of the exact same genes, which are also in similar locations as chimpanzees and other apes. Just one viral group, HERVWs, I think, have about 205 out of 211 similarities. And again, that's just one virus group. There are several of them. And the basic idea is that since there are only so many places for these viruses to insert, first of all, the likelihood that it's going to insert into a gamete is very unlikely in the first place. And then you have to have the specific species of virus and specific strain of that species, and inserting in the same location of the same genome in two separate animals, humans and chimps. And we do have very similar number of similarities with other apes and monkeys, and those same similarities create nested hierarchies, which look exactly like the same nested hierarchies that are produced by other genomic analyses and by anatomical analyses. And so this on its own indicates a common ancestry. But as I started off talking about, the likelihoods of these things happening in two separate animals are inconceivably high. And, it could be as high as 5.8 times 10 to the 1,418th. I'm sure that number will be disputed a little bit, but basically the lowest you can get it is still an astronomical number, up to like 3 times 10 to the in the 60s, which is still, I don't know what that would be. That's like somewhere between like quintillions and Google. Google is like a number with 100 zeros after it. So it's just an incomprehensibly high number. And the only way that you could get around this indicating common ancestry is if God put them there, except they are viral infections. So this would look really bad for the idea of intelligent design, because God's updating the system with a really inconvenient way to update the system, and for some reason he's updating both chimps and humans in the same way, in a really inconvenient way, in a very unlikely way, astronomically unlikely. So there's really no way to get around the fact that these are inherited, and the layout of the suite of viruses, viral remnants, is exactly the same and could not happen unless it was inherited by a common ancestor. And so that's where we're at. So I will cede the rest of my time to discussion.
All right. Thanks for that intro, Taylor. Looks like you had about 7 minutes to spare, so we can add that to the end. For now, we'll hand it over to Donnie for his intro. And whenever you're ready, Donnie, start on your first word there. Go ahead.
Okay, much appreciated. And let me just get my screen shared here. Brandon, I think there are some people in the chat just asking who they tag for questions. So that'll be Brandon at Brandon for your questions. Okay. So let me get my PowerPoint up here. And let me know when. How's that look? Coming in, Brandon? Audio, everything?
Looks epic.
Yes, the epic showdown. Okay, let me get my timer going as well. I was writing things down like crazy there, and time flies by. Okay, so 12 minutes. All right, the epic showdown. Endogenous retroviruses, ancient viral infections, or created units of DNA function. This is my roughly debate number 97, and I look forward to this discussion. So, what exactly are ERVs, or endogenous retroviruses? An ERV is a stretch of DNA found in your DNA that, according to evolutionary theory, and as Taylor pointed out in his opening statement, got there when one of your ancestors was infected by a retrovirus. What is a retrovirus? A retrovirus is a special type of virus that inserts its genetic material directly into a cell's DNA. Right here, what is an endogenous retrovirus? They are transmitted vertically, right, rather than horizontally through the germline, and are thus inherited by a successive generations in a Mendelian manner. Reverse transcriptase. This is important right here to understand when it comes to this topic. Okay, because retroviruses, however, use a slower, stealthier approach. After entering the cell, the retrovirus uses an enzyme called reverse transcriptase to turn its RNA into DNA before making its way to the nucleus. Once in the nucleus, it inserts its DNA into the host's genome. Now, when these retroviral genes make their way into sperm and egg cells, they can become a permanent part of a species' genome. Proponents of common descent, like Snake was right here, will frequently claim the existence of these ERV sequences are irrefutable evidence for common ancestry, since they can essentially act as a historical record of infections suffered by our past ancestors. And I've got notes here. Snake pointed out that essentially there is no way around this for the creationist. Essentially, they'll say ERVs, the existence of these shared ERV sequences across various taxa, precludes separate ancestry. So this is going to be a fun discussion. Now, these small pieces of DNA found in the genome are recognized by various signatures. These signatures reflect similarities found in exogenous retroviruses. And because we share these ERV sequences with the primates, as Taylor went over in his opening statement, we must have inherited, this is their only conclusion, these from a common ancestor in the distant past. Proponents of common descent essentially believe endogenous retroviruses are inherited, are the ancient remnant, here we go, ancient remnants of past viral infections that have integrated into the genomes of living organisms. And that's why in my opening slide, the question is, are these really the ancient remnants of past viral infections, or are they created units of DNA function? They assert that these ERV sequences are clearly the remnants of viruses. They don't question it. To them, there is no debate. When we look to the properties of the ERV sequence, we can see that they are found in retroviruses. Evolutionists will also point to, and Taylor mentioned this in his opening statement as well, they'll point to the nested hierarchy that these sequences fall into. And we'll definitely be discussing that specific line of argumentation as well. And again, ERVs that are shared across species are evidence for common ancestry to the evolutionists. Now, I do want to go over kind of some of the details and some of the basics before I continue into kind of the meat of this topic, as it is my goal for people to be able to follow tonight's debate. And at the end of it all, see exactly why ERVs are not really good evidence for common descent. And as a matter of fact, they are amazing evidence for biblical creation, as we are going to see. But more specifically, the design diversity model. Now, when a retrovirus becomes a permanent part of a species' DNA, according to the evolutionists, it becomes an endogenous retrovirus. This is important to understand. Scientists call it endogenous because it is inside of us from birth. A retrovirus is not passed on genetically or vertically, is referred to as endogenous. If it's passed on vertically, it would be referred to as endogenous. But if it's passed on horizontally, we're looking at that which is exogenous, okay? And it is the retrovirus, as I pointed out, that is passed down genetically that we would refer to as endogenous. And that's what we're discussing here tonight, because the organism will be born with this viral DNA. Now, the human genome contains thousands of ERV sequences. As I touched on earlier, these stretches of DNA match sequences found in retroviruses. And to the evolutionists, this is why there is no question, as Taylor pointed out in his opening statement, that these are indeed the ancient remnants of past viral infections that have been passed down, essentially. And the question is, though, how do we know for sure genes with similar sequences to virus genes actually came from viruses? And again, to the evolutionists like Taylor here, it's the important properties of the ERVs themselves that tell us these DNA elements actually originated from retroviruses. Notice on this specific slide, the structure of ERVs match modern retroviruses, for example, HIV. On both ends of the retroviral DNA will be two identical sequences known as LTRs, or long terminal repeats. In between the LTRs, we find the GAG, the pol, which codes for the reverse transcriptase we were talking about earlier, and the ENV, or envelope protein, which codes for the envelope that makes up the body of the virus. These structures are common in ERVs and retroviruses. Remember, again, it's important. ERVs are assumed by the evolutionists to be the ancient remnants of past viral infections. Evolutionists consider these to be genetic fossils that point us to common descent. Advocates of common ancestry would say that the chances, and Taylor pointed this out in his opening statement, that the chances of two ERVs being inserted at the exact same location in separate organisms are very small. They will argue that the chance of a human and a chimpanzee being infected in the exact same spot by the same specific type of virus is far less than 1 in 10 million. And to them, as Taylor put it, this is highly unlikely. The more shared ERV sequences then that we find, the more unlikely it becomes that these were inserted what? Inserted independently. Okay, so in a nutshell, why do evolutionists believe ERVs are a good line of evidence for common descent? One, the sharing of similar ERVs at similar locations in different genomes, plus the nested hierarchical distribution of ERVs themselves. Two, the properties of the ERV itself. We covered that. And three, the examination of shared mutagenic discrepancies between the long terminal repeats of LTRs of shared ERVs, essentially the structure itself, okay, that also form a nested pattern of distribution across various organisms. So here's some important questions we have to answer. Why are there ERV sequences shared between the genomes of organisms? And two, if viral-like sequences in the genomes of organisms are functional, which I am going to argue is the fact today, why do they bear similarity to viral genetic material? So as we know, the entire junk DNA paradigm has been overturned, essentially, which is confirming evidence for the design diversity hypothesis, which suggests that God would have front-loaded the original created kinds. This includes Adam and Eve, with created diversity, okay, created nuclear heterozygosity, essentially, as well as functional DNA elements, such as these ERV sequences, pseudogenes, allele sequences, so on and so forth. So as you can see here, pseudogenes necessary to sustain healthy life processes in the cell, lines, signs, introns allow for alternative splicing, ERVs. ERVs are what we're focusing on today. So they function now in antiviral function, tumor suppression, gene regulation. We are going to get into all of that today. Now, the question is, why do ERV sequences resemble viral genetic material? And this is a common response from the evolutionists after we show them just how functional and essential these retroviral-like elements are. And we have an easy answer. Okay, one of the major functional roles of these viral-like elements, these ERV sequences, is that they have a purpose in the innate immune system. They play an antiviral role. They're important DNA elements that work greatly in the immune system of their host. The way they mechanistically exert their antiviral effect has to do with their sequence similarities to viral material. Without the specific nature of these ERV sequences, they could not do the important job that they do. Thank God they resemble viruses. Thank God they have similar properties to exogenous retroviruses. Their functional roles depend on these similarities. This is not evidence that they are the ancient remnants of viral infections. No, the properties of an ERV sequence is necessary in light of their essential roles in aiding in the immune system and fighting off viral infections. In my last 2 minutes here, I want to go over some of the more amazing functions found in these types of sequences. It has recently been reported that ERVs can act as DNA regulatory elements, as long non-coding RNAs, and as triggers in the innate immune system. Okay, and this isn't just creationists making this up. Notice this from the secular literature. ERVs, HERVs, human-specific ERVs, appear to play important roles in physiology, fetal development, and human evolution. Notice this, if the accidental, notice this, to the evolutionists, it's all an accident, right? Blind chance. Infection of a mammalian ancestor by an exogenous retrovirus had never occurred. The placenta and the mammals that produce it, including humans, would have never existed. Beneficial role of human endogenous retroviruses. It has been suggested as mediators of normal biological processes such as cellular differentiation and regulation of gene expression. And in my last 40 seconds here, I mean, we could go over function after function. Here's the one that I was specifically referring to earlier, how endogenous retroviruses protect us from viral infections. They not only regulate cellular immune activation, but may even directly target invading viral pathogens. And that's exactly why they look the way they do, because this is necessary in light of this one of many, many functional roles, which points us to the design diversity hypothesis. And I'll wrap it up there as I've got 1 second left, and we will touch on some more of that evidence a little bit later. Thanks so much.
Excellent intros, gentlemen. I'm sure that's going to raise a lot of questions, if not within the audience alone, amongst you guys. So at this point, we can move on to the rebuttal stage. We're going to do 8 minutes apiece for that. Whenever you're ready, Taylor.
Yeah. So, we heard a lot about how ERVs contain beneficial functions, and therefore they had to have been designed. But this is not exclusively expected by creationism. So one of the ways that, for example, ERVs can help in cancer suppression is, well, one, it's kind of a double-edged sword because they actually can cause cancer. Some of them cause cancer and don't help at all. Some of them cause cancer and then help our immune system identify cancer because they're producing viral antibodies, which the body has. Depending on the age of the virus of origin, our bodies may have already been used to these viruses for thousands, even millions of years. So the antibodies are easily identifiable by the immune system. And so I guess to kind of recap, an ERV could interrupt certain genes, which can lead to higher susceptibility of cancer. But then those cancer cells that are expressing that virus now look like virus infected cells. And so our immune system can kill it. And so, of course, that can work out accidentally pretty easily. It seems pretty obvious how that's kind of a self-canceling property of a cancer-causing element that can also tag itself as something other than human. So the immune system often doesn't understand that cancer cells need to be killed because they have the same proteins as all your other cells. So the immune system can't tell the difference. And so if there's some foreign element that is causing the cancer, then it's more likely that your immune system is also going to recognize that foreign element. So it's completely understandable. It's not like a wild, convenient coincidence. It's very obvious that your body would recognize it as a foreign element. So just the mere fact that it has a beneficial function does not really tell us anything about whether it was created or it was infected. But the structure of it can tell us whether it was infected, because the ERVs contain elements that are completely unnecessary in from an intelligent design perspective to insert into the genome. So you wouldn't need the pro-GAG and the EMV genes because you could just incorporate those genes into the normal genome and put them in an exon, and they would get translated just as our normal proteins do. And those could trigger viral antibodies. And you also wouldn't need, and from an intelligent design perspective, it's also a terrible design because a lot of these ERVs are interrupting genes, which can cause cancer. So, so basically the functions that these ERVs take are going to be infection related, immune related, and gene regulation related, which is exactly how retroviruses function as they infect us. So none of these beneficial functions are unexpected or conflict with the idea that they are historical infections or how biochemistry works in general. Viruses and infections aren't inherently bad for you. In fact, you could not survive without, I don't know the exact number, but I think it's in the thousands of species that infect your gut that you can't digest without, or that infect your skin that keeps away worse infections. And as a parasitic or infectious pathogenic or a pathogen, they don't want to kill their host. So the most successful pathogens actually work with the host. Same with the symbiotic biological relationships. You can suck the blood out of your host, but if it kills it, you run out of your food supply. If you're hardly noticeable, or if you actually provide some kind of benefit, then your host is actually going to want you around. One example is the birds that they're not exactly parasitic, but they clean out the teeth of the crocodiles. The crocodiles don't eat the birds that hang out in their mouths because they clean out their teeth. So these kind of relationships can develop naturally, and it's going back to ERVs. It's not very well. It's not. It's close to zero. The probability that, in fact, in one of my calculations, my calculator literally couldn't calculate that low and just gave me a 0 for the likelihood. So the idea that God would put these things in our genomes that were not in the ideal locations, but they were also in the exact same locations as other species, and the fact that he would use an update to an operating system instead of just put them in our genomes in the first place is really odd, and doesn't jive with intelligent design at all, because in fact we can come up with better designs. So the fact that they're in the same locations does indicate that it was a historical infection shared by two different species, not because of the similar sequence, but because, well, they're identified by the similar sequence, but again, similar species, similar location of a similar gene, well, almost exactly the same. And it's the fact that this isn't going to happen just by chance. I had another point, but I derailed my train of thought. I'll get it in a minute. But the idea that God would use ERVs to infect us is, even if he's putting them in the correct location or whatever, even if there's not a better intelligent design, ERVs are one of the worst delivery mechanisms for retroviruses are one of the worst delivery mechanisms for updates to genetics, because they're not necessarily going to get into the gametes. And we even have therapeutics that are based on retroviruses, but the fact that they're so unlikely to get into the reproductive cells is a major problem for this intelligent design method. And I think that should be about 8 minutes.
You are exactly correct, man. 8 minutes. Pretty good job. A lot of good points there, man. I think you brought a little more heat in the rebuttal than you did in your opening, huh? All right, we're going to hand it
That
over
was
to
planned.
What's that?
That was part of the plan.
There you go.
I think we all kind of know the topic, so.
Turn up the heat on him, man. He needs it.
Yeah.
Okay, well, here we go. Let me start my timer, 8 minutes. And the openings were brief overviews. And here we go. Let's get into the fun, because I am pumped. So Taylor's arguments here, essentially in the opening statement and the rebuttal itself, boiled down to a starting point and basic assumption that these viral-like sequences are really the leftover remnants of past viral infections. Just so stories aren't going to show us empirically how non-functional endogenous retroviral-like sequences can go from something that is non-functional to critically functional in determining cell types, aiding in the immune system, embryological development, and so on. Okay, because he pointed out that it's not just one beneficial function. Okay, he what I've written down is he said, a beneficial function, one single beneficial function, can certainly come about through co-option or evolution. It's not just one. Okay, there are massive amounts of ERV-like sequences that are aiding in transcription and in so many important functional roles. And something important to note is that retrotransposons, okay, they are mobile genetic elements, and we know that they're often referred to as jumping genes because they can actually move around the genome, jumping from one location to the next in the DNA itself. They can literally move from one chromosomal location to another. And the ability of a retrotransposon to move around the genome allows for what? It allows for genetic variation when mobilized. So here's another important functional role of these various classes of retrotransposons. And that's why there's a creationist model called the VIGE hypothesis, which stands for, that acronym stands for variation inducing genetic elements. And the fact is, in what this model proposes essentially, is that a lot of these bad viruses have actually originated in endogenous retroviral sequences within the genome rather than outside. And we can actually make viruses in our genome. This is an important point because in our cells, we make all of the parts that viruses make. We have all the ingredients and properties to make viruses. We make protein shells, we make DNA, we make RNA, we have DNA and RNA copying enzymes. It is possible that many viruses came from within the genome rather than outside. And this had to be the case anyways, because a retrovirus requires a host to replicate. So what came first, the virus of the host? It makes sense, and it solves this dilemma and paradox that evolutionists face in terms of the origin of viruses in general, specifically retroviruses. I'd like to ask Snake, what's your explanation for the origin of these retroviruses that require a host in the first place? Okay, so it is possible that many viruses came from within the genome. They were made from cellular parts. As our cells are packaging things in various different ways, a few accidental changes during the packaging process can actually make a bad virus. And it sounds like viruses came from the human genome rather than vice versa. So his response here in my notes, where he says that a lot of these ERV elements are detrimental, cancer-causing, disease-causing. Well, that's exactly what we would expect through mutation accumulation, through accidental errors in the genome may result in fully working endogenous retroviral-like sequences, altering to the point where they may cause disease and may cause cancers of all sorts. But the fact is, one of their main roles is the fact that they play an antiviral role. And because of this, they require what he was saying aren't required, like the GAG gene, the POL gene, the ENV, okay, the LTRs. No, these are all functional properties of the overall endogenous retroviral unit, okay? And there's a lot of latent genetic information in our genome. We understand this. And many of these retrotransposons actually have a gene promoter in them. And so if they're stuck in, let's say, one place in the DNA, and within this place in the gene, they can turn on a gene. Okay, they regulate. And if they move, that gene gets turned off. So you have these signatures of integration, not because they are being integrated from the outside, but because they are actually moving around within the genome itself. Okay, so if this mobile element moves, that gene gets turned off. And what this tells us is that as jumping genes are popping in and out into the genome in different places, they can literally turn things on and turn things off. This is evidence for forward thinking, which points us back to a forward thinker. And I want to share screen and kind of just demonstrate a lot of what I'm saying here in the number of papers that I have made for everybody. Right here, ERVs are retrotransposons, a type of transposable element that spreads throughout the genome via a copy and paste mechanism. And there's a different class that can do it through a cut and paste mechanism, okay? Retrotransposition. This capacity allows ERVs to make copies of themselves that in turn insert themselves elsewhere in the genome. And that's why you'll often see them being referred to as mobile elements. All right. The ability of transposons to increase genetic diversity. This isn't coming from creationists. Here's an article in, you can find it on Nature. Together with the ability of the genome to inhibit most transposable element activity results in a balance that makes transposable elements an important part of evolution and gene regulation in all organisms that carry these sequences. So again, jumping genes. And here's something that's interesting about the function of of ERVs is that much of the evidence for the function of Ts comes from the growing realization that many transposons are highly conserved among distantly related taxonomic groups. So if what we're looking at is high levels of conservation, that would mean they must be there for a reason if they're not being hit with all of these mutations that essentially could be damaging, because maybe we're looking at a sequence that's nothing more than genomic leftovers, genetic baggage. No, sequence conservation suggests functionality. And this is an important point, guys. ERVs frequently have important immune functions, and they should not be presumed to be junk DNA. This defeats both the junk DNA or junk ERV argument against the design of the genome. It also challenges those who want to use the supposed junk status of ERVs as an argument for common ancestry. So the last thing I want is that the LTRs, okay, they're there for a reason. They have the capacity to exert regulatory influence as both a promoter and enhancer of cellular genes. We went over the fact that what we could be looking at in terms of bad viruses is viral escapees. And last thing I'll say is this argument from co-option, it's imagination. Okay, I'm going to end it here with a simple question for Snake, I guess, as we move into the open discussion. After this, he can either choose to answer it first or not. Show me empirical evidence in a lab, a technical paper today. It's not just one or two functional roles in these ERV sequences. Show me a non-functional ERV sequence going from non-functional to something critically functional in the genome. And that's 8 minutes, so I'll yield there.
Excellent. Perfect timing. So now we're going to go into this 30-minute discussion, and I guess we can kind of tail off right where Donnie left it if you want to. Snake, do you want to address that question? You got anything else you want to bring up?
Well, I guess I would offer like a slight correction. So, so the GAG and the envelope proteins are potentially useful. They're not necessarily useful. But the pole section and the long terminal repeats are basically just virus specific, and so the human genome is capable of translating the GAG or pole regions, or the ENV regions, independently of the LTRs, long terminal repeats, and the pole, which is the reverse transcriptase, right? And so there are markers that show that these things are in fact viral, and so and are not necessary as intelligent design elements. So, like, do you accept that some of these ERVs are in fact of viral origin?
Well, some interesting points that you brought up. So to kind of work from your first point is that, yes, when we look to these ERV elements and the properties essentially that make up the ERV sequence, right, the structure of it, the LTRs on either side, your GAG, pole, and envelope protein, essentially. Okay, these are characteristics and signatures of exogenous retroviruses. Okay, they have similarities, but that's what's so fascinating about the structure of the ERV sequence itself, is the fact that one of their roles, Taylor, is they have the ability to protect against the exogenous retroviruses that supposedly they are the result of, right? You have this viral infection, and essentially it gets passed on because it invades the germ cells, and it's passed on vertically rather than horizontally, and so it becomes endogenous. It comes from within. Okay, the offspring are going to have this viral DNA in every single one of their cells. And essentially, that's where these ERVs came from, according to the evolutionary story. But the ability of these ERV sequences to actually protect against this Okay, the fact that ERVs block the ability of the exogenous retrovirus to infect in the first place and to become an endogenous retrovirus tells me that, and you can see this in a number of papers I can screen share later. For sake of time right now, I won't. They perform viral mimicry, and they require the specific structure and properties, okay, including the GAG, the pol gene, the ENV, in order to carry out this ability. And another ability as well is involved in tumor suppression, where they also, we can get into that a little bit later, where the structure and property of the ERV sequence itself, these similarities, are necessary. They're a required design feature to carry out the job that it does in disrupting retroviral insertions and in tumor suppression. So I don't think the properties that make up the ERV or ERV that are also similar to exogenous retroviruses is a problem for the design hypothesis. What are your thoughts on that? Go ahead.
Well, yeah, I definitely want to get into the topic of discussion. But as for the question, do you accept that there are retroviral infections that result in these ERVs, even if you're not accepting any of the beneficial ones?
Well, that is a good question. And it doesn't seem out of the realm of possibility that an exogenous retrovirus can invade our cells and invade in just the right way and in the right spots for it to be passed on vertically, essentially. So it doesn't seem to be out of the realm of possibility. So how many have actually occurred, have gotten past one of these functional roles of the ERV sequences in general to kind of prevent that and stop that? I'm not sure, because as far as I know, we don't really have any observable evidence, unless you can present that, where we have endogenous retroviruses. We have a new the virus infecting and then becoming endogenous and fixed within the population. Because from my understanding, and respond to this, what we're looking at is genomic fossils to the evolutionists. And so this is a historical record of common descent. But we're not actually seeing the endogenization, I guess you could say, occurring today. Go ahead. What are your thoughts?
Yeah, so like, how would you tell the difference between a retrovirus mimic and a legitimate endogenous retrovirus?
Well, right, that's a good point. But
because, well, if I could add a little bit more, so we do know what retroviruses look like when they infect cells, and they have this exact same structure. And the only difference would be that they occur in gametes, which are reproductive cells. And as long as that doesn't kill off the fetus, which is one form of selection, if the mutation or the infection occurs in a highly inconvenient place, that thing's just not going to even be born to pass on its genetics. So, but yeah, we use retroviruses for therapeutics. So we know how they look when they infect cells. It's just a difference of which cell they infect. So going back to the question I asked, is there a way to differentiate a virus mimic and a legitimate ERV?
Right, so that's a good question. I would say the majority of what we're looking at in terms of these, quote unquote genomic fossils that evolutionists such as yourself look to and compare across different groups, different species that fall within a nested hierarchical pattern, as well as the mutations that occur in these LTRs, these LTR elements. I would say that the vast majority, if not all of those, are created units of DNA function. They are these variation-inducing genetic elements that were front-loaded, and in a way we can tell is whether or not they are functional. That's a direct prediction of the design diversity hypothesis, that if God front-loaded various types of functional units in living organisms, that would include these ERV sequences, ALU sequences, we would predict function, and we would predict a lot of function, not just one or two. So when we find an ERV sequence that is functional in, let's say, embryological development, or beneficial in, like right here, human endogenous retroviruses have recently been suggested as mediators of normal biological processes such as cellular differentiation and regulation of gene expression. So I would say, okay, confidently, this is a created unit of DNA function, given its functional role. And for you to demonstrate that, no, this is the ancient remnant of a past viral infection, you would show how it's actually possible empirically for this type of DNA unit to become functional in determining cell types, gene expression, embryological development, in the immune response. So that would be my criteria, is looking for function. And if we have an ERV sequence, here's the last thing I'll say, and then take as long as you want. If we have an ERV sequence, okay, that is functional, let's say in the immune system, and then we have another ERV sequence that maybe we haven't really tested, so we don't know what the function is yet, but they still bear the same signatures and similarities, I would say, okay, this is, I would predict this is also a functional ERV element, but what's its exact function? We still have to test that, which is nice, because this is where testable predictions that are falsifiable come into play, you know? So respond to anything, but again, I'd like to ask you, given the criteria, what would be your best example of a non-functional retroviral-like element or sequence going from non-functional to something functional in the immune system, in embryological development? I mean, that paper that I showed in my either opening or rebuttal, the authors, which are secular, they admitted that without this endogenous retrovirus, we wouldn't be able to reproduce. It's required for embryological development. So, Snake, go ahead.
So, to kind of correct something, retroviruses have function from the moment of infection. So that you're never going to find anything that goes from non-function to functional. You might have kind of a change of function or a shifting of responsibility, but we can talk about that once we're fully on the subject of function. But so it looks like you're saying that they're identical in structure, and you can't really tell based on their genetics whether they're from an infection or not. It's just whether or not it has a beneficial biological function. Is that how you're telling the difference?
I would say the criteria in terms of determining what is a created unit of DNA function versus what is a retrovirus. For example, when we looked at the Phoenix virus experiment, okay, that's a paper you've cited before. That's a great experiment where they took cells in a petri dish and subjected them to various mutations. They produced a virus. Well, that goes back to what I was saying in my rebuttal, that one creationist hypothesis titled the variation-inducing genetic element hypothesis, which suggests that retroviruses, exogenous retroviruses, have originated from functional ERV-like sequences. And at that point, they can escape the genome, cross species, and of course become damaging, disease-causing. And I think that has to be the case anyways, the nature of retroviruses. So if we're looking at what you would say is a genetic fossil, a dead virus, that is what I'm saying is a created unit of DNA function. If we're looking at an exogenous retrovirus, then I would say that is what probably originated from these functional ERV sequences. And I'd like to see where a retrovirus, an exogenous retrovirus like HIV, assists in embryological development or determining cell types. I'm not aware of any research suggesting that these retroviruses, before they integrate and are passed down vertically and become endogenous, I'm not aware of any papers that suggest that they already contain the necessary information to assist in embryological development. So go ahead, Snake.
So you did say that it is possible for at least one beneficial function to come about by infection and co-option. So
I wouldn't say co-option.
Okay, I mean that's what you said, but you're
Well, I
you're
was saying,
welcome to clarify.
well, I don't want to, I just want to clarify. I don't want to say that it's impossible. I'm not going to sit here and say, no, it's impossible for an exogenous retrovirus to infect a germ cell, enter a sperm or egg, and then be passed down vertically. And the offspring now has an I'm just saying, I'm not going to sit here and go on record saying that's impossible. Maybe it's possible. If one of their roles, these ERV sequences, is to prevent that, well, we know we have DNA repair enzymes that are formulated to prevent mutations, but mutations still occur. So I'm not going to say it's impossible, but it's not like we have overwhelming evidence in the lab or a series of technical papers showing this happening. I mean, you can present, if you can present that, I'd like to see it, but we don't see that as far as I know. Go ahead. I just wanted to clarify.
Yeah, as far as cells that are infected with retroviruses, they can reproduce and they still have the retroviral elements in them. And so as far as co-opting a function, like I said, not all viruses are going to be harmful. And some of them are going to be harmful in the right way. So there are certain genes that we could benefit from having down regulated, and if the virus infects those genes, then that's going to be beneficial because it's damaging a bad gene or a gene that might need to be turned down or interrupted in some way. So yeah, what's your response there?
Well, a few responses and a few questions I would have is, if what we're looking at here, then these thousands and thousands of ERV sequences that essentially are the ancient remnants of past viral infections, okay, that have been passed down over millions of years. How in the world did these ancestors survive to reproduce, especially this invasion of deleterious exogenous retroviruses? How did they survive this invasion of all of these viruses? How was this not deleterious to the point where, for one, selection would just remove the infected group from the equation, and two, how would this result in fixation, since we're looking at fixed ERV like sequences? Go ahead, Snake, if you want to answer those.
Well, the first step, obviously, we survive all the time, just as we are now, getting infected by all kinds of things. As far as deleterious ones, I explained there is a selection filter where, just like any mutation, if it's too detrimental to the organism, it's not even going to gestate, or it will gestate badly, and if it's born, it will die soon after or not be able to reproduce. So there is a selective filter based on how harmful the inclusion of the virus is. So I mean, half of all pregnancies don't even implant. Half of all fertilized eggs don't even implant, and there are, I don't know how, I think it's in the like dozens of millions of sperm cells per ejaculate. They're all going to have different mutations and potentially infections. So if the infection is bad, the sperm cell will be unfit. It won't even get to the egg. If somehow it gets to the egg, then it probably doesn't have an infection bad enough to slow it down. If it if the infection doesn't slow down the sperm cell, it might have an adverse effect on development, and thus that genetic element will not be passed on. As far as co-opting a function, we see this in a lot of different organisms where two different genetic elements or anatomical elements can serve the same function, and they can lose one of these and get along fine because there's a redundancy there. So if the ERV is providing a similar regulatory function as a regulatory function that the organism already has, it is now free to have that ERV mutate, and this happens a lot where ERVs might be slightly harmful, but we can sequence them and see that they've lost some of the pathogenic elements of them, so that they're not harmful. If you have that redundancy, you can have mutations in either the ERV or the previous regulatory element, and you'll still have the same regulation. So there's no theoretical or functional barrier to the co-option of those ERVs, even as essential functions.
Okay, so I'd want to respond to quite a bit there. First thing I'll say when it comes to the answer to the question about how these species could essentially survive just mass invasion of virus after virus. I do believe it, unfortunately for the evolutionists, does require a lot of just so storytelling. We know that this hypothetical invasion of viruses occurred millions of years ago in order for them to be passed on. And after the human to chimpanzee split, we have the similar ERV sequences because the human line, chimp line went different directions, but retained these endogenous retroviruses essentially. But my biggest counter to that is the paper that I showed earlier in my rebuttal that demonstrates we're looking at a lot of conservation, a lot of conserved sequences where these ERV elements haven't really been hit with a ton of mutations. And so this would suggest, and this was from a secular technical paper as well, this would suggest that we're looking at genome-wide functionality, essentially, of these ERV sequences. Now, of course, through mutations, recombination, and other accidental changes, these could destroy the functions of some of these ERV sequences. Also, we know that a lot of these ERV sequences are only functional. They're expressed during different stages of development, okay? And after they are expressed and essentially they do their job, they are then turned off or suppressed in their function. So if we were to test that, let's say in the life of a specific organism, if we were to test that, and even through genetic knockout, we were to snip it out and there was no immediate effect, well, that's because either one, we're looking at redundancy, two, we're looking at an ERV sequence that is turned off. It's only expressed under certain conditions, certain environmental conditions. When it comes to tumor suppression or antiviral infection, they're only called upon essentially in light of those circumstances. So there's a lot of ERV sequences, unfortunately, that evolutionists would say are non-functional, but they haven't done enough, I would say, analyses or even knockout experiments to know that for a fact, since we know for a fact that a lot of these ERV sequences are only functional under certain conditions. And again, when it comes to the co-option, you know, here, for example, far from being junk DNA, the pervasive retrotransposons that populate the genome have a powerful capacity to influence genes in chromatin. A new study demonstrates how the transcription of one such element, HERVH, can modify, notice this, can modify the higher order 3D structure of chromatin during early primate development. Here's the last thing I'll say, and then take as much time as you need, Taylor. Okay. In the mouse genome, there's a certain class of retrotransposon that if you snip it out, okay, you remove it, the mouse is developing, you remove this retrotransposon and the mouse stops developing, it dies. It's because, again, it requires that functional retrotransposon to develop and live. Okay, so I don't think your answers here are adequate, that retroviruses have some innate functionality that when they, in fact, integrate and are passed on vertically, okay, through the germ cell lines, they can eventually become not just functional in a low functional sense, but literally critical, as in that one paper I showed earlier, demonstrated that no, if it wasn't for this ERV sequence, there wouldn't be mammals. It's literally required for the existence of mammals. So I don't think you're adequately answering this question, because to me, the question could be adequately answered with just a single technical paper that actually shows empirically in a lab, hey, listen, here's an ERV element that we thought was useless, but through a series of mutations or co-option events, it went from something that was dead to something that is now critically functional in embryological development. Can you provide anything like that, Taylor? And go ahead, take your time.
Yeah, so they're not functionally dead when they infect. They're functionally, well, there's a debate over whether viruses are alive or not, but the retrovirus, as soon as it infects, is functional. All of its viral elements are functional to create viruses. So basically, to gain a function, parts of it have to die so that they're not viral and pathogenic. But as you said, they can affect regulation. And that's really not hard to explain, because mutations affect regulation all the time. They can affect cellular division. And viruses do have self-regulatory elements in them. It's not like once they're inside of your body, or once they've inserted into the genome, that they just continuously replicate and replicate and replicate. There are tons of diseases that can go dormant, and there are certain environmental conditions that will then trigger them to turn on. And so they have an ability to interact with the cell, whether it's interrupting it or tagging it for being activated, which are not, they're completely unremarkable as far as non-designed elements go. And like I said, if there's a redundancy there, the original function to that can be lost, and it can look like, oh, the ERV was the only thing that was ever regulating this, when it's perfectly capable of having just been a redundancy that is left over.
Well,
and yeah, go ahead. I'm going to be listening, but I'm just going to grab something real quick.
no worries. So again, the example that I pointed to, where you have a class of retrotransposon that, if knocked out, if removed, the organism stops developing, it dies. I mean, we're not just talking about the known properties or function, I guess you could say, of exogenous retroviruses. Okay, the evolutionist is saying that these ERV sequences that we see, where the debate lies, are these really the ancient remnants of past viral infections or created units of DNA function? You guys are the ones purporting. For example, I had a bunch of papers here up from, let's see if I can get to them real quick, from the secular literature, where the best you're going to get, for example, here, if in the past HERVs were mistakenly considered as useless elements of the human genome, DNA junk, which they're admitting that in the past, this is what they assume. So one question I'll have, if you want to keep it to memory, is Do you have any technical paper or any example of an evolutionary scientist predicting function before function was found? That's a question I'd be interested in hearing an answer to. Today, some are recognized as conferring biological advantages. In fact, in some cases, HERV genomes have undergone a process of positive selection during evolution, being exploited by the host to benefit important physiological processes. This is a paper from 2020. This is the best you can find. I've read through dozens of papers, and they don't actually, they'll say, well, these are engines of evolution, positive selection, kind of like what you're doing. And apologies if this sounds aggressive. I just think it's a lot of fanciful storytelling, a lot of informative gloss, and essentially imagination. Like, just imagine, an exogenous retrovirus that essentially is infiltrating in order to do damage to the host. The exogenous retroviruses we know today, like HIV, for example, nobody wants that infection, right? And again, thank God we're seeing it in these populations of koalas, where these ERV sequences are actually now being called upon. And thank God for their structure and properties, the LTRs, the GAG, the POL, ENV, which the evolutionists, that's their best argument. They always say, like you're saying, why do they resemble exogenous retroviruses? They literally require these similarities. They require these signatures in order to do their job when it comes to fighting off bad viruses. They have to have that structure in the first place. And so the VIGE hypothesis, at least, would say, okay, they look the way they do because one of their many functions is to fight off bad viruses. So the last thing I'll say, and then take your time, is I don't feel satisfied with the answers provided, not only by you, just in the papers that I'm reading, where it discusses these functional roles. I never actually see empirical evidence for how this co-option happened or how this evolution happened. They just say it did because positive selection, engines of evolution, so on and so forth. So, obviously you can disagree with me, but I don't feel like an adequate answer has been provided to how these incredibly important functional roles can come about. Taylor, go ahead, take your time.
Yeah. Well, first of all, how much time do we have left?
Brandon, you might be on mute, and I'm.
You got the 30 minutes is up in 10 seconds, but we can add the extra 7 minutes that you didn't use in your opening. So if you guys want the extra 7, you can have at it.
Yes, please. So as I said, viruses are not necessarily bad for you, or they could. It's all completely context dependent. So they can be completely neutral. They could be, in fact, we use viruses to infect and kill cancer cells because they can be selective like that. Some viruses kill off bacteria, but don't infect humans. Or viruses might have a disruptive effect on harmful genes or mutations that we already have in us. So the regulatory nature of retroviruses already explains it in any evolutionary way that they can regulate things. The only thing, if there's a dependency there, this would seem to indicate that it was not based on the evidence that it was an infection, would seem to indicate that it was a part of a redundancy that we've lost. And we see that such as there's a species of amoeba which has a sister species, and there's one species of bacteria that infects both of them. One of the cousins is killed by the bacteria. It's pathogenic in them. The other will die without that same exact species of bacteria. And so there's very little difference between them. But one of them, they're the same biblical kind, but one is entirely dependent on something that it did not have before getting infected by it. So, and this is based on redundancies. So it had something in its genome that was able to keep it alive before. One of the pathogenic bacteria infected the amoeba, spread throughout the population because it was not as pathogenic as a variant that it's related to. And then the amoebas that could tolerate these bacteria basically lost their original function, and now you can't take the bacteria out without killing them. But they originally had this function. So we observe this process happening. We know it's possible in viral regulation. So,
Snake, if
and
I
as
could, I
yeah,
just wanted to make sure you answered the question and still continue on as long as you need. One of the questions I asked in my last response was, did evolutionary scientists predict function before function was found? Do you know the answer to that?
as far as the literature says, it's usually was not known. So it's an investigation. They discovered viral elements. A lot of people assumed because it's an inactivated virus that it wouldn't be that important, but then we discovered that it was. So
Isn't that just
I mean, we're not.
retrofitting the data though?
No, that's just investigating what it is. Like there's no one's claiming anything outside that it's like if we're investigating the genome, we find these elements that look like viruses, and eventually we found out that some of them have beneficial functions. So that's not retrofitting any data. The only thing that we're
Well, what would be, if you could, are you, like, if you could screen share or present us with, based on what you're saying, okay, what would be the best technical paper or lab experiment that you can provide myself or anybody on the fence, let's say in the audience, that would show us and kind of make the light bulb go off, where we're like, okay, we can see how, because if you notice this paper here, ERVs appear to play important roles in physiology, fetal development, and human evolution. This is not a creationist paper, by the way. If the accidental, notice this, the accidental infection of a mammalian ancestor by exogenous retrovirus had never occurred, the placenta and the mammals that produce it, including humans, would never have existed. And I want the audience to pay close attention to that. Without this endogenous retroviral-like sequence, we couldn't exist. Humans would have never existed. These beneficial consequences can explain why these HERVs have been fixed into the genome instead of being eliminated over the years. The most parsimonious explanation for me is, okay, these are necessary, these are essential, these were designed and created. But for you, I just see, unfortunately, and I understand you'll disagree, I do see a lot of kind of just so storytelling, maybe this, maybe that. But why in 2022 don't we have a technical paper documenting some kind of lab experiment where this endogenous retroviral-like element was subjected to a series of mutations or whatever that actually led to a highly essential endogenous retrovirus? Yeah, go ahead. What would be your favorite technical paper just to kind of throw at us, demonstrating what you're saying in terms of the co-option?
you mentioned the Phoenix virus, so we can make them. We know that these elements are telltale signs of a viral infection. So at some point, yes, some regulatory function is beneficial. That's
Well, what did
not
the Phoenix
contrary to
virus
any.
experiment demonstrate, though, for the audience sake?
It demonstrated that it's a virus of viral origin.
Well,
So,
firstly,
but
how is
even
that
then,
demonstrating the co-option of these functional roles in embryological development?
well, we know that this is possible. I can cite the amoebas paper that we know that redundancies with this type of thing occurs. It's extremely rare. Like you said, there's one of these out of thousands of these. So it's not going to be easy to evolve this in a lab. But we do know that they are viral elements. We do know that they're a result of an infection, and we've kind of lost track of the fact that there are hundreds of these that are shared with chimps. So this is, again, since we know that they're viruses, statistically impossible
Well, I just
have the
want to make
same
sure that the
suite.
audience, obviously I disagree with that. We don't know they're viruses. I think we've demonstrated that they're not. But I understand you believe that we know they're viruses, but the only reason you conclude that is because of their properties, the structure, the LTRs, the gag, the pole, the
They're
ENV.
viral exclusive properties, right?
But I've already pointed out that the structure, the makeup, the properties of the ERV sequence are required for one of their many, many roles, which is fighting off exogenous retroviruses, working in the immune. If they did not have these similarities to extant retroviruses or exogenous retroviruses, they couldn't do their job. So I don't understand why you believe this is evidence that they're ancient remnants of viral infection.
So that would be something like the envelope protein, but the rest of the telltale signs are not necessary because you mentioned viruses being bad for us. We can't have an immune system unless we're exposed to viruses and bacteria. So our immune system literally needs examples for it to learn. We produce antibodies that go after all kinds of things, and this is a process of miniature evolution. It's called somatic hypermutation, produces tons and tons of different mutations, and then the ones that work survive and proliferate, and then that's the immune system that you work with going forward. And if you encounter more, there's that. And then you say that these are, if they're not retroviruses.
If I could, okay, I have to point out that no, every property of the unit, the ERV unit. For one, a lot of what we're looking at in terms of these sequences are just LTRs, just functional stretches of DNA. You know, we only find very few full endogenous retroviral sequences. I think you understand that. But if you're looking at the LTRs, the gag, the pole, the ENV, it's all functional for the unit. Okay, it would be like saying, well, you know, a human being is not a functional unit. It just has some functional parts and other parts that are not functional. Well, that doesn't make any sense. The ERV unit itself is made up of properties or ingredients, I guess you could say, that are necessary for its functional roles, including the fact that they jump around, retrotransposition. They can jump around the genome, okay, leading to genetic diversity, turning genes on and off. They require their structure to even do that role. So I just wanted to say, I think you're wrong on that, Snake. Go ahead.
No, because you don't require them to have retrotransposable elements. They stay in the same place. That's part of the reason that's part of the whole argument, because they're in the same place. If they were hopping around completely, that would...
Well, they can move around. There's class 1 and class 2. They can either move around through a copy and paste mechanism or even a cut and paste.
Yeah,
they're jumping genes, right?
But the fact that they're found in the same locations is one of the pieces of evidence. So that's not a functional element. That you don't require it to jump around for it to express envelope proteins, that's one of the ways that it's beneficial to cancer, is it starts producing viral specific particles that the immune system can recognize. But it doesn't need an entire it doesn't need the LTRs to express those envelope proteins. And in fact,
the LTRs have a different function.
But as you said, not all of these are complete retroviral sequences anyway. They have function and get along fine without the entire suite of retroviral tags.
Right. A lot of what we're looking at in the genome are just LTRs. And notice here from PubMed, endogenous retroviral LTRs as promoters for human genes. In this review, we summarize known examples of LTRs that function as human gene alternative promoters, as well as the evidence that LTR exaptation has resulted in a pattern of novel gene expression. You can read through this all yourself. My hand and my foot and my heart and my lungs, okay, everything that makes up me, okay, the unit that is a human being, has different functional roles, does different things, just like the ERVs made up of LTRs, or you just have isolated or solo LTRs that are just functional stretches of DNA, critical in gene promotion, things like that. Either way, what we're looking at, regardless of the property, it's functional and necessary. And it contradicts this idea that these are the ancient remnants of viral infections. Again, I haven't really seen the Phoenix virus experiment. That doesn't work. That just showed us exactly what we've been predicting, that retroviruses have originated from within the genome. So they subjected an endogenous retrovirus to a series of mutations and revived a virus. Well, that's exactly what we're saying based on our VIGE hypothesis. So that doesn't work to answer the question. I just think this is a question that evolutionists really do struggle in explaining, and I understand why. Go ahead,
Snake,
Taylor.
Yeah. Why don't you go ahead and take the last word on that? Address what he said there. Take your time, and then we'll move into your guys' closings if you want.
Yeah. So the entire suite of retroviral parts is not necessary for the beneficial function of it. Some LTRs are beneficial, some ENVs are beneficial, some GAGs are beneficial, but not the entire thing. You can cut it up. And as you said, some of them are missing some of those parts, but those are identifiable tags that show us that they are viral elements. And we can give you that it's a retrotransposon that originated in the genome. It doesn't matter. Well, I'll finish that thought, but so even if so this is with the LTRs example that you gave, this is exactly what we expect evolution to do. Is we have promoters, we expect that new DNA elements are introduced or moved around that can change the regulation of certain genes or change the composition of the proteins of the genes that get made. And this is not news. It's just news that some of the viruses are confirmed to have functions. So that's exactly how we expect evolution to work, is to co-opt new pieces of genetic information. But as far as we can give you that, they're not even the results of retrovirus infections, but simply created transposable elements. I'll give you, God created these pieces of DNA, but if you're going to go with their transposable elements, there's no mechanism for them to transpose to the exact same place or any very specific place. So the fact that they transpose in the exact same location, in the exact same genes, in the exact same location of chromosomes with the exact species, is still evidence that they are an inherited suite of a pattern that comes from a common ancestor.
Well, before you go into your closing, and maybe answer this in your closing, you understand that a lot of these jumping genes, these retrotransposons, they do have, it's not entirely random. They have preferred integration sites, as in maybe, between this gene and this gene is going to allow for the essential function that it requires, or this will allow for the for genetic variation when they jump around. So they have so it's not just completely random. A lot of what we're looking at is non-random. And so that's why we would see them jumping into a lot of similar spots. Maybe save that for your closing. You have 5 minutes.
Sure.
Yeah, let's move into that. All
right.
Like you said,
Am
Snake, you're going to get 5 minutes. Yeah, you'll go ahead and go first.
All right. So yeah, I calculated being extremely generous and only, again, this one viral group, the ERVWs, HERVWs. If we're saying that there's 211 that we have, And we're going to be extremely generous and say that there's only 211 possible places where it can insert, which is way too generous to be realistic, but let's go with that. Let's say that each virus can only go to one of these 211 places. And let's say that each has a 50% chance of going there, because yet, like you said, there are preferred sections, but there are still millions of them. But we'll reduce it down to 211 and give it an extremely generous chance of 50%. We still have a 3.5 times 10 to the 64th chance of it occurring the way that it does. And so there's that. Plus, it happened exactly the same way in chimps. To the 205th. So that's still statistically, it's a bigger number than the quintillions. So even if we're being extremely generous and saying that it's a retrotransposon that's originating in the genome, and it has exactly as many places as there are and no more, and we're giving it a 50% chance, it's still statistically about zero likelihood that this is going to happen the same way in both chimps and humans. There's the fact that it just so happens to coincidentally match the exact same phylogenetic trees as is represented by genetic barcoding of any other gene, or and the exact same phylogenetic tree as happens with morphology. That's also a coincidence, because it's a suite of traits. That can't be a coincidence, is what I mean to say. Because again, there's hundreds of different insertion sites that could be anywhere. That could be functional anywhere. We know that could be functional anywhere. These glowing jellyfish in my background, we just took their fluorescent protein and we can just stick it in pretty much anything and make it glow, and it doesn't really matter. You can't just stick it literally anywhere, but you can put it on different chromosomes, things like that. So the location is not important to function. So this is an extremely poor design for God to have programmed, knowing full well that people like I and 99% of all of the best scientists are going to see the evidence in apparently the wrong way. I'm not sure why he's going to make the decision to give a design that's apparently deliberately confusing. And I've got about a minute left, I think. As for functional elements, they're all related to things that viruses are already capable of. They behave exactly how we expect these signals to behave. It's not out of the realm of probability to expect regulatory elements to have deleterious, neutral, and beneficial effects. You can just throw them randomly into things and they'll have beneficial, deleterious, and neutral effects. This happens with viruses, and so that's not a problem. The fact that we observe in many species redundancies that can then be taken up, that you can then lose one of those redundancies and still have a necessary function, that explains every functional element that it required functional elements. So yeah, I think that just about wraps it up.
That does it for your 5 minutes, Snake. Excellent closing. Very good. It's been a lively debate. Tons of points made. So, Donnie, whenever you're ready to give your 5-minute closing, man, have at it, and I'll start your timer.
Okay, I appreciate it. Let me start my timer as well. Lots to cover in just 5 minutes. So no, it's not bad design. It's actually a perfect design. Okay. These elements are essential. They're critical to immune responses, cell stress responses, embryological development. I mean, during this debate, I must have shown at least over a dozen technical papers verifying exactly what I'm saying. And Taylor, unfortunately, he didn't provide even one technical paper showing empirical evidence for how these elements can evolve such as essential functions, including embryological development. I mean, that one paper I showed from the secular literature literally admitted if it wasn't for this retrotransposon, we wouldn't exist. How is that bad design? They're admitting we wouldn't exist without it. That's good design. I like existing, and I think Taylor likes existing as well, okay? We share these elements with other forms of life like the chimpanzee simply because of common design, since guess what? Chimpanzees also require elements to help with embryological development, determining cell types. Did you know chimpanzees are also made of cells? Yeah, believe it or not. Gene expression, gene regulation, okay, aiding in the immune system, tumor suppression, so on and so forth, okay? So they require these elements as well. Were we just supposed to expect to find them only in humans and not in any other form of life? That sounds kind of bizarre. And Taylor tries to counter the fact that these endogenous retroviral sequences have incredibly important and essential roles that if you knock out a certain retrotransposon, the organism dies. Again, that is good design. But he looks to this Phoenix virus experiment where scientists took human cells, mutated the DNA ever so slightly, in the endogenous retrovirus, and it turns out that an extinct virus was revived from a DNA sequence that was found in the human genome. That's exactly what we would predict, because as I've pointed out several times in this debate, okay, RNA virus, retroviruses require a host to replicate. What came first, the host or the RNA virus? I asked this question to Snake a couple times. Obviously, the host, these had to have originated within the host, and we would suggest that they originated from functional ERV sequences. And that makes sense. We can actually track back every single exogenous retrovirus, given the genes and the properties it has. We can track them back to the host that they originated from. Okay, and there's many creation scientists, one specifically named Dr. Pierre Terborg, I think is how you say his last name, who has done just that. And there's been no counter. Okay, so the Phoenix experiment does not help. It actually helps our model that suggests that a lot of these viruses came from ERVs, came from the human genome to begin with. There's a paradox. There's an unsolvable problem with those that believe in deep time evolution. Where did RNA viruses come from in the first place? Okay, so no, this is completely compatible with our model and does not answer the question, the important question: are these really the ancient remnants of past viral infections, or are they created units of DNA function? Okay, one function that we kind of touched on slightly is that when the cell is under stress, okay, transcribed ERV elements gives the cell the appearance, notice this, of being invaded by a viral infection, which as a result targets the cell, targets that specific cell, more destruction by the immune system. And some tumor cells can invade detection of the immune system, but this mechanism, which is mediated by what's called, it's a super protein, okay, P53, is a way for those tumor cells to become detectable by the immune system, as a way for the immune system to clear these tumor cells from the body. And researchers, they're working on various drugs that will stimulate P53 activity and the transcription of ERVs as anti-cancer therapy. Anti-cancer. These ERV sequences are playing a role in tumor suppression through viral mimicry. Guys, the properties and structure of the ERV sequence is required. It's a necessary truth for these ERV sequences to perform their roles. Notice here in the last 40 seconds, why would a creator produce functional genomic features that so closely resemble an endogenized retrovirus? Okay, we've seen this in real time. I've shown many technical papers. The structural and functional features of the pre-existing ERVs, their capacity to copy themselves and move throughout the genome, are precisely what make these ERV sequences so useful. Their capacity for retrotranspositioning affords these sequences the means to disrupt the endogenization process of invading retroviruses. They literally they operate as antiretroviral elements. They must, and I'll say it again, they must resemble endogenized retroviruses. They have to have these similarities to extant retroviruses, or else they couldn't do their job. End of story. Slam dunk. ERV argument for evolution is, I think, dead. It's a fatal blow. So I'll yield there. That's 2 seconds left. So that's my closing statement. I appreciate it.
Excellent timing again as well. Well, pretty good debate. I mean, I almost tailed off a few times not paying attention to the timer because you guys are bringing up some good points, a lot of good questions, a lot of good information. Now we get to move on
Technical
to
subject,
the real fun part, right? The audience Q&A, and we've got quite a few here. Looks like we got about 17 and a half minutes left till we hit the 2-hour mark. So we'll see how many we get through. I'm going to scroll down and we'll jump right into the super chats. First off, George Bond with the $10 chat. Sweet chili chicken burger for lunch. Not bad, George. That sounds pretty good. Alec Cox with a $10 chat. He threw in some random stuff here, but his question, how long does it take a cell to divide in a human? Number 2, what is missed in that division? And I'm assuming this is pertaining to something Snake said.
Yeah, I'm not sure what is missed in that division. I'm not sure what that's supposed to mean.
Me neither. How about any thoughts on the first part? How long does it take a cell to divide in a human? Alex asked.
It's a couple hours, 12-ish hours, depending on the cell. 12 hours.
Excellent.
I'm not sure what the second part is supposed to mean.
Me neither.
You're getting a quiz, Taylor, apparently.
Yeah.
Right. All right. We'll move on to the next one. Question for Donnie. Has anyone seen a modern viral insertion into the genome that hasn't caused serious health problems for the host, something similar to HIV?
Right. So when these modern viruses, these extant viruses like HIV, when they invade the host cell, okay, they end up it can be passed on horizontally, but not vertically, like the endogenous retroviruses, and they result in disease. They're deleterious. They're damaging. And these retroviruses are very interesting because I pointed it out in my opening statement is that they use a very specific enzyme called reverse transcriptase to transcribe. They're made of RNA genetic material. Okay, so they use this enzyme to transcribe the RNA in reverse back into DNA. And the virus uses another important enzyme called integrase to integrate its genetic information directly into the host's DNA. Okay, and if you are infected with one of these extant retroviruses like HIV, for example, there's another one, the exact name of it I can't think of right now, but it can actually result in lymphoma, various cancers. Okay, these are deleterious, these are damaging. But what we see in terms of the endogenous retroviruses that are tightly regulated and integrated into the genomes of living organisms, they function in the embryo. They function in determining cell types. They function in gene expression. They have antiviral properties. They function in tumor suppression. They aid in transcription, act as promoters. I mean, I can go on and on and on. And that's not deleterious like these extant retroviruses. So I yield there.
And what was the question again?
The question was, has anyone seen a modern viral insertion into the genome that hasn't caused serious health problems for the host?
Modern, yeah, I'd have to look into exactly what it was, but yeah, most diseases are not going to be fatal. And you were mentioning reverse transcriptase a lot, which is one of the hallmarks of a viral infection, which is not going to be required by something, even if even those elements that are required to mimic a virus for cancer regulation is not going to require reverse transcriptase. That's just going to tell us that it's actually from an infection.
Any last thoughts on that, Donnie? You want to move on to the next one?
Yeah.
Okay, so it's my question. I'll give the last couple thoughts. So again, yeah, a lot of diseases that come about. Okay, not every single like a single point mutation can kill an organism. Okay, well, you know what, that organism's removed from the equation. Okay, selection's dealt with that. Selection is all about differential reproduction. Who's having the most babies? Yeah, most diseases are not detrimental enough to fitness where that person experiencing the disease can't have kids and then pass on their deleterious mutations with it. Like if I lost both my hands and both my feet, let's just say even through a mutation, that sucks. But it's not going to prevent me from having kids. Okay, so yeah, many diseases are not going to prevent reproduction. And the last thing I want to say is, again, when it comes to the reverse transcriptase, the GAG, the POL, the LTRs, the ENV, whatever, okay, every single property is essential for what these do, specifically the transposons, how they are mobile genetic elements, and they can move around the genome, jumping from one location to the next in the DNA. They can do this through a copy and paste mechanism or a cut and paste mechanism. And the structure, including the reverse transcriptase property of these ERV elements, are required for their many functional roles, this specific one in generating genetic variation, which is what creationists predicted. I'll yield there.
Excellent. I hope that satisfied the questioner there. We'll move on to the next one. I think this one is for you, Snake, pertaining evolution. They say, so if evolution is the result of random, unguided processes, why should we expect to see any kind of a pattern at all?
Because it's not. It's not unguided at all. It's guided by chemistry. It's guided by selection pressures. So, I mean, and we can prove that these exact mechanisms can create functional elements, whether we're looking at computer simulations of random mutations and then being subjected to selection, which can cause functional code or functional body parts if we're simulating 3D body parts. All these exact same processes work. We can also observe it at the genetic level with actual organisms. But again, it's not unguided. So I'm not sure what that's usually a straw man, calling it an unguided process, because it's not.
Okay, Donnie?
Yeah, if I could, these patterns that we see, let's say specifically the nested hierarchical patterns, the fact that we share more with other primates, let's say we share more with the chimpanzee than we do with a lemur. We share more with even the lemur. We can be grouped together with the chimpanzee and the lemur because we share more in terms of morphology, anatomy, physiology, and genetics than we do with a dog or a fish. I mean, this is expected, of course. Just stand back and look at the organism. You're going to predict more similarities in genetics with the chimpanzee than with the dog. But even when it comes to these patterns that apparently, according to the evolutionists, are expected and predicted, from the common descent starting point, we see inconsistencies all the time. There's something called incomplete lineage sorting, where depending on what gene you look at, depending on what genetic marker you look at, you're going to get totally different trees. Look at the Y chromosome, for example. The Y chromosome between the humans and gorillas is more similar than the Y chromosome between humans and chimpanzees, there's a break in the so-called hierarchy. And even when it comes to these endogenous retroviral like sequences, there's inconsistencies in the hierarchy as well. For example, here's a paper, a HERV, whenever you see the H, human endogenous retroviral, K provirus in chimpanzees, bonobos, and gorillas. These observations provide very strong evidence that for some fraction of the genome, chimpanzees, bonobos, and gorillas, notice this, are more closely related to each other than they are to humans. Okay, but the bottom line is we do share more endogenous retroviral-like sequences with, let's say, the chimpanzee than we do with a creature that we share less in terms of morphology and anatomy with, because we share more in terms of anatomy, morphology, physiology. So of course we're going to share some of these ERV sequences with them, especially if they function again in the embryo, determining cell types, gene expression, so on and so forth. That to me points to common design, explaining the pattern far better than evolution. I'll yield there.
Last word, Snake.
Yeah, so that seems to be an inconsistency that you would expect similar morphology to have the exact same genetic elements. But then you point out places, isolated places where there are certain breaks in the phylogenetic tree. But that's not really how phylogeny is done. It's done with a suite of traits, not just one single trait. And the Y chromosome specifically is one of the most fastest mutating, it is the fastest mutating chromosome we have. But it's not really, the arguments, at least with ERVs, is not about strictly about the similarity. It's about the fact that has gone basically unaddressed completely, which is that regardless of where you think they originate, these are elements that came to rest where they are in the genome via either infection or transposition, which is statistically impossible to end up the exact same way in two different species like that. And the fact that we can predict how much, that's just an added cherry on top, I guess.
Excellent. All right. So before we move on to the next question, let me just say that Logical, Plausible, Probable is having an after show, and that is exclusive for cool people. So if you're not cool, don't even think about going over there. High probability of dumpster fire, and I am a fan of probability theory, even Bayesian probability, which puts the resurrection at a probability of 0.97. And I would say the probability of dumpster fire might not be as good as the resurrection. We'll put LPP's dumpster fire like 0.96. But make your way on over there after this if you want to. Let's get on to the next question. I think this is for both of you guys. Okay, it's good. Only losers won't come to the after show. All right, so question for both of you guys. Do ERVs in similar areas of coding sequence, because of similar pressure and similar purpose, ERVs seem to be no more evidence of common descent than of common design? Same pressure, same region.
Who wants to start?
Well, you could make certain arguments that it is, I guess, agnostic for things like function. Yeah, it could go either way. But again, the thing that really differentiates it is the viral elements that, viral exclusive elements that we know are from viruses, or if you prefer, transposable elements. Like you said, sometimes there are more or less parts of the virus where the fact that it looks like an ERV is important, and it's expressing itself in cancer cells so that it can be detected and will target cancer cells. This is to be expected. If there's an infection, an added genome can tag things to be differently, to look differently. But other things like the mammal required placental ERV, it doesn't have to mimic an ERV. So there are certain parts of the genome that are not necessary but still mark it as an ERV, and that's how we're able to identify them as separate parts. These aren't just random pieces of the genome that we're deciding had to have come from viruses. They have specific tags. Yeah, all the papers you're referencing have been reviewed by evolutionary biologists, and they come to the conclusion that they are evolved. Like the one you quoted about mammalian ERVs is about the evolution of retroviruses. We should go into detail about that study. There should be around 2. But I guess I started off my statement saying there are functional elements which could be agnostic, which could be designed. The thing that is not agnostic to this debate is the likelihoods that these elements are transposed to the locations that they are is basically 0. So, and we haven't really addressed that at all.
Okay. Well, I appreciate that response. First thing I want to add is I'm choking. That's the first thing I want to add is the evolutionists keep saying that these are expected, but then they admit that they never predicted the function. So how are these things expected if they never predicted the embryological developmental requirements essentially of these retrotransposons? And I saw someone in the chat. Yeah. Apparently the technical way of saying ERV is ERV. They're ERVs or ALU sequences, ALU. But oftentimes it is easier just to say ERV, endogenous retrovirus. But yeah, so the evolutionists never predicted. And I'll go on record saying they retrofitted the data. And of course, the authors of these papers are not going to conclude, okay, special creation. I mean, that's off the table. Okay, they are required. Sure, they can question some details of the evolutionary story, but they can't question the bigger picture. They can't conclude, well, okay, this must mean they're created units of DNA function. Are you kidding me? That paper wouldn't be published. That's why they say things like motors or engines of evolution, positive selection. That's not telling us how an endogenous retroviral-like element can go from what they once assumed was junk to something incredibly functional in the immune system. I mean, I find this so funny. So apparently we were invaded by exogenous retroviruses, which today endogenous retroviruses fight against, protect us against. So these exogenous retroviruses got passed down vertically, became endogenized, and eventually evolved the ability to fight off that which they were before. I mean, that sounds to me kind of bizarre. Okay, a lot of this is just fanciful storytelling. It's a fairy tale. And here in this question, if I'm understanding it correctly, I want to urge creationists. Taylor does have a point. If we are saying that these are actually the result of ancient viral infection, some creationists hold that position. And I wouldn't recommend that because Taylor is right. We share a lot of these sequences or even fragments with the chimpanzees. And so it's very unlikely that we independently experienced the invasion of these viruses in similar positions. Okay. One, given the amount of locations in the genome, is somewhere like 1 in a million. So if you go into 200, 300, it's even more than that, of course, because that's not what the evidence suggests anyways. The fact is the mutations that we find in the structure of the endogenous retroviral elements, like the LTRs, yeah, that can be due to environmental pressures. We know mutations are not as random as once expected or predicted. No, mutations are essentially, a lot of them are non-random. Okay, we also know that evolutionists assume all DNA differences are the result of mutations over time. So a lot of these DNA differences are, quote unquote mutations in the properties of the ERV, like the LTRs that form these nested hierarchical patterns, may not even be the result of mutations. They may be functional differences that are required for the respective organism. So anyways, I could talk all day, but I'll just say, you know what, as a creationist, we should go with what the evidence suggests. And the evidence suggests that these are created units of DNA function. If a creationist wants to say that these are the result of ancient infections, or even infections thousands of years ago. Now the creationist is stuck in the same position as the evolutionist. How do you explain the functional roles? How did those, get adopted essentially? So I'll yield there. Good question.
Anything you want to add to that, Taylor?
So in that paper you were talking about, there are conserved sequences of the ERVs and non-conserved sequences. So they're using that to track basically the lineage of a certain ERV, which, I mean, there's different levels of this. So there's the shared ERVs, and then within those shared ERVs, there are even further differences that aren't seen in the conserved domains. And the point there is the highly variable regions are essentially not functional because they're so highly variable, especially LTRs. But
well, actually, Taylor, since that was a question for both, can I just respond to one thing and then
I have one more thing to respond to.
Sure,
as far as what was it? I kind of derailed my train of thought there. So yeah, go ahead. If I can, I'll get my train of thought back, but you can go ahead.
Apologies. I almost forgot what I was going to respond to. Okay, so the conserved versus non-conserved regions. Okay, when you find these conserved regions, let's say we just stick with ERV sequences. If we find an ERV sequence in, let's say, humans and chimpanzees that's nearly identical, or conserved, as you could put it, evolutionists say, well, we have these conserved sections of DNA in humans and mice because they're essential, they're important, so selection has essentially conserved or preserved them over time. But here's the thing: an ERV sequence in humans that say has to deal with determining cell types or regulating genes, but specifically we're focusing on similarities in morphology or anatomy, that ERV sequence is going to be very similar anyways. And that could be interpreted as conserved. But then non-conserved regions, that doesn't mean that it's non-functional. It just means there's also some parts or aspects to the gene expression pathway found in humans that's a lot more dissimilar to chimpanzees. So we would expect that the evolutionists would say those are non-conserved. No, they're different because humans also have a lot of differences with the chimpanzees. And so those functional differences would reflect in the sequences as well. And the DNA differences in those sequences, the evolutionists might say, are due to mutations over time. But maybe those differences are actually there because they're reflecting functional differences between humans and chimpanzees. So that was kind of a mouthful, but I'll yield there. Go ahead, Taylor.
Yeah. So, the thing I lost my train of thought on was the idea that I'm losing it again. I'm going back. What were you saying? The being able to you said that we're just kind of accommodating the data. So I guess I wanted to clarify. So it wasn't predicted necessarily that ERVs had a specific function, but it's not unexpected. Like, they weren't surprised or caught off guard that ERVs had the specific functions that they did, because they already knew that viral elements had that function of they can regulate themselves, they can disrupt genes. In fact, that is one of the causal elements of why so many retroviruses can cause cancers, is because they interrupt cell division regulatory genes, apoptosis regulatory genes, which is programmed cell death. So when we confirmed we're not going to be able to predict everything about the genome because of basic Darwinistic mechanisms. So there's always going to be something to discover. But it's always in line with what we know about what the functions that viruses are capable of have. So we're not seeing things like I don't know, like a blood coagulant or like an antifreeze gene coming off of the these things that apparently look like ERVs. We're seeing virus related functions. So yeah, and just to kind of liken it to crime scene investigation, we're not going to say, well, it looks like the victim has been stabbed with a knife, but he could have just been born with the knife in his chest. We're going to say, okay, we know knives can get there by the process of stabbing. Same thing we're doing with the ERVs. We know that we know that it looks exactly like this when retroviruses insert themselves. So, and a lot of the functions are not related to needing the entire retroviral suite of traits. And that's why there's such a highly variable region. Yeah, go ahead.
It
So-
seems like that question alone could have been a whole nother debate.
Exactly. Yeah.
Well, I just want to ask one simple question and then we can move on. Are you going on record saying that exogenous retroviruses, they can accomplish many crucial functions, such as regulating gene expression, cell differentiation, and embryological development? Is that your position?
They have the ability to regulate or disrupt genes that they insert into.
Disrupt, we've seen, but we have not seen an exogenous retrovirus integrate itself into the genome, be passed on vertically to be essential in embryological development to the point where we could say, wow, if it wasn't for that retrotransposon, we couldn't exist. Show me a technical paper on that. Send it to me after, or, I'm going to call a bluff on that one, Taylor.
But we have seen is that their ability to regulate the genes that they're part of, or inside of, or bordering.
So we've observed the turning on and off of genes, these exogenous retroviruses involving themselves in the gene expression pathway. You're saying we've observed this?
Yeah, that's what they do.
Okay, so what's a technical paper that shows an exogenous retrovirus infiltrating the cell and and then adopting a function where it actually helps in gene regulation, cell differentiation, expression, things like that? Like, we've observed this in the lab, you're saying?
Well, when you're going highly specific, laboratory experiment that includes a beneficial, necessary retroviral insertion. I can look for one for you. We can have around two. But as far as the functions, regulatory functions are, and as far as the fact that we can tell what DNA elements are inserted and transposed, we using the evidence that they are in fact retrovirus infections, we can tell that certain viruses or certain ERVs necessary for development were the result of infections. As far as us accomplishing this in the lab, I'll have to search for one for you. But
well, we see these ERV sequences that you say are genomic fossils, right? Brandon's laughing, never ending. We see that they function in turning genes on and off. That's what gene regulation is, essentially, okay? And we know that gene regulation is incredibly important in development. And these genes are often turned on and off based on the presence or non-presence of these ERV-like sequences. So we see that they function that way. But we don't see exogenous retroviruses invade the cell and then evolve that function. That we don't see. So it sounds like to me you're seeing the functions in the endogenous retroviral-like sequences and assuming, well, the exogenous retroviruses may already have the built-in capacity for that function. Is that essentially what you're saying?
Well, we know that they do have the built-in capacity to regulate.
To regulate the host genes in helping with development, turning genes on and off. We know that.
If it happens to affect a gene that is involved in development.
Well, let's see that.
Sure.
And I'm just asking, let's see that. Let's see. I want to, because all I ever see is engines of evolution, positive selection. We don't see, okay, here's an empirical example, an observable example of an exogenous retrovirus integrated into the cell, being passed down vertically. And now, wow, look at this. It's critically functional for the organism. I don't believe we see that. Maybe I'm wrong. And we do, but I would just have to have you prove that to me, is what I'm saying.
Yeah, but what I'm saying is that's an unreasonable standard of evidence, because we already know that redundancies and dependencies can evolve, and we already know ERVs
But that's not
can
an
insert.
answer, though.
We already know that they can regulate, and we know where we know that these elements come from.
I think we'll save that one for round 2. Huh?
Round 2. Yeah, it's a good topic, and this has been enjoyable, Taylor. I appreciate it.
We could make it just on that mammal paper.
Well, here's a question that might lead you into that about mammals and endogenous retroviruses inserting themselves. The question states, if we see similarities in monkeys, would we then find some of the endogenous retroviruses to be in other mammals like whales?
We do find
directed towards you, Taylor. Yeah, go ahead. Sorry for cutting
we do
off.
have some similarities with whales, yeah, as far as ERVs go. Like you said, a lot of these are mammalian specific, which all mammals share, and there's a phylogenetic tree, a nested hierarchy, that shows the same relationship as is predicted by evolutionary biology through morphology.
Yeah, the ERV sequences are abundant in jawed vertebrates, mammals, obviously, and primates. We're talking specifically here about the primates and how these ERV-like sequences do fall out into a nested hierarchy, as well as their mutations. A lot of the times in the structure of these ERVs, they also fall into a hierarchy. But here's the thing, again, going back to the beginning of the debate, is what we're looking at in terms of these so-called genomic fossils. Are we really looking at the ancient remnants of viral infections? I'm saying no. Or are they created units of DNA function? That I'm saying yes to. And we can predict function. Evolutionists admit that they never predicted it, but they say over and over again that we expected it. Okay. Well, if you didn't predict it, and essentially you're retrofitting the data, and all their papers never actually say how these functions were co-opted or came about, they just make the claim. They just assert co-option, rapid evolution, engines of evolution, so on and so forth. But they don't actually give an empirical reason for a creationist to believe, okay, non-functional properties of the endogenous retroviral element or unit, went from non-functional to something incredibly functional in determining cell types, gene expression, so on and so forth. And again, the follow-up question by the evolutionists is, well, why do they resemble exogenous retroviruses? Why do they have, LTRs and the GAG gene, the pole gene, and the envelope protein, for example? And it's like, we just had a paper in either 2021 or 2022 that I showed earlier that demonstrates that these properties and the structure of the ERV is required for its purpose and job. I mean, that's perfectly compatible with the creation model. And the evolutionists can reject it and say, well, no, we were invaded by a virus, and then the virus became an endogenous retrovirus as it was passed on vertically, and then it evolved the ability to fight off that which it used to be. I mean, to me, that just sounds ridiculous. When it comes to the hierarchy, of course, if these are created units of DNA function, okay, this should be basic to anybody, then of course we are going to share more. For example, humans and chimpanzees, we are going to share more of these ERV sequences with the chimpanzee than we're going to with the mouse or a whale Okay, because we already share more with the chimpanzee in terms of morphology, anatomy, physiology, and genetics. A sedan is going to, by definition, share more features with the SUV than it is with a bicycle or a plane. Just stand back, look at the structures, and anybody can predict that. So the nested hierarchies that we find are agnostic. What's not agnostic is the function. That is my position. I am making the claim that DNA function can help differentiate between the models. Ancient viral infections created units of DNA function, repredict function. Evolutionists, although they've had to retrofit the data, they predict these are evolutionary leftovers. Okay? And so they would predict non-function. They'll still predict most of them are non-functional, even though that logically has been overturned. So I believe the DNA function and the lack of response from the evolutionists in explaining the DNA function is the fatal blow to their interpretation of the endogenous retroviruses. I yield there.
Taylor?
Yeah. So yeah, the nested hierarchies are agnostic until you get to transitional forms that connect all the separate hierarchies. And like what you were saying about the cancer regulation ERVs that need more of the viral elements, that's a completely different mechanism from the mammalian developmental ERVs that do not and do not need to resemble a virus. That's related to regulation, not immune development. And then the final point, I guess, is just prediction versus expectation. So it wasn't, we didn't predict the functions, we investigated the functions. So, and then we didn't predict non-function. It was more just a lack of, we don't know what they do. We're not predicting what they do. So they investigated what it does. But all functions were within expectations of what viruses function as. So I don't want people to confuse expected virus function with a prediction that a specific ERV is going to function in a specific way.
Interesting. I do have a question from Dustin. It's a little tough to read. I don't know if you can see this, Donnie. I asked him to rephrase it in the comment section a while back, but let's see what you guys can make of this. He says, where does the line lie as far as an inherent genetic trait and a new something that develop? I know that sounded kind of choppy, but again, he says, where does the line lie as far as an inherent genetic trait and new ones that develop? I don't know what that says. I can't. For example, alcohol was once thought to be passed down, but not every ancestor had access to alcohol. Does that make any sense to you guys?
The line between inherent genetic trait and new ones that develop. As if I try and understand that as far as the evolutionary perspective, there isn't a line because new traits that develop are derived from inherited genetic traits. Not sure. Maybe the enzyme to metabolize alcohol is what he's talking about.
It's possible. I don't know what a WON is, the WONS. I don't know what WONs are. So maybe can you make sense of that, Donnie?
He's talking about the difference between genetic traits that we all share and traits that have been regionally adapted.
Yeah, I don't see him in the chat anymore. Maybe he left. So if you got anything to say on that, Donnie, feel free. If not, we can skip that one.
Yeah, let's move on to the last one or two because we're coming at the 2.5 hour mark and I'm getting pretty exhausted at this point. Very interesting technical debate. So
this is the last one here from Bubblegum. Bubblegum Gun it's a little tongue in cheek, but this is more of a statement to the both of you. He says, how convenient that these ERVs found just the right spot to insert themselves to later in the future become necessary. Such foresight from that virus.
Yeah, so that's not, as I've explained, that's not really how it works. So the fact that something is necessary is not an indication that it was ever the only necessary element or the only part of the genome that served that function. We know there are biological redundancies. We know the mechanisms by which you can gain a redundancy and then lose that redundancy, but the part of the genome supporting that function changes because there is a redundancy. It's like if you have, I don't know, two different ways to support, like my computer is supported by a couple of books right now. I'm putting it up high. If I put another book in there and I take another book out, it's still supported. So it's not the second book having the foresight to be necessary. There are a lot of ERVs that are not necessary, and in fact are still harmful.
So much that could be said there. So I'm going to hinder myself from a monologue, but I think Bubblegum Gun nails it there, where it must be convenient that essentially these deleterious exogenous retroviral infections invaded the genome, invaded the cell, invaded it in the right spot to be passed on vertically and eventually become an endogenous retrovirus. The offspring would have this viral DNA in every single one of its cell. And then eventually it also adopts, co-ops, evolves the essential functions necessary for humans to even exist. Okay. Essential in embryological development, snip out that certain class of retrotransposon, and although the mouse is developing, bam, you snip it out, it dies. Okay. And evolutionists, when they talk about redundancy, they'll often talk about it in a different way than we would. Okay. Redundancy is evidence of forward thinking. Anybody who does computer code understands that. Okay. Or let's say a spare tire in the back of your car. Well, to the evolutionist, that might be redundant. Like, why is that there? I've already got 4 tires and they work just great. But you know what? If the environmental condition arises where you get a flat tire, thank God you got that spare. Okay. But what's funny about that is once you use that spare, okay, that's it. You're even redundancy can only go so far, because mutation, like treading on your tires, every time you go out driving, you are ruining the treading, and eventually you'll need new tires. Now it's there, it's redundant, it's there for wear and tear, but it can only last for so long. Okay, any computer coder can tell you redundancy is evidence of forward thinking. The evolutionists, though, they look to redundancy, and essentially they give evolutionary mechanisms a mind. Because they'll say, well, these neutral mutations, which we know is false, we know they're nearly neutral, effectively neutral, slightly deleterious. They build up over time, and they become like a hidden reservoir of genetic change, where at some point this quote unquote redundancy or genetic baggage can be called upon through mutations, neo-functionalization, co-option, whatever, co-option when it comes to the ERVs. And they can become beneficial. Okay, now this hasn't been observed specifically with the ERV sequences. We haven't observed the types of functions that we see. The evolutionists never predicted it, and they'll admit, no, we never predicted it. You know, we used to assume that these were junk, useless, and now we know that they are functional in tumor suppression, gene regulation, antiviral function. Okay, again, I want to point out, it sounds silly to anybody who thinks about it logically that these viruses invaded the organism and then eventually evolved the ability to fight off that which they were. It's like a robber breaking into your house, but deciding to live there for some reason has an agreement with the people that live there and own the house at the time, and decides to just have a change of life. He repented, and now he's going to fight off future robbers from breaking into the house. It's ridiculous. And a lot of this, it's storytelling, it's imagination. We want the empirical evidence, we want the technical data, and we just focused on ERVs tonight for the most part. But I mean, we can talk about all sorts of function for what was once assumed to be junk DNA. Nucleoskeletal hypothesis is fascinating. I talked about it with Dr. Fazrana in my 2-part series with him. These so-called junk DNA regions are acting as a mutational buffer. They're maintaining the 3D structure, which is amazing. It's like this just came about for no reason, because of the buildup of neutral mutations, regulate histone binding, gene regulation, the pseudogenes code for functional proteins. So I'll end it there. I just think that the whole model of these neutral mutations building up to the point where they can be called upon for beneficial change or novelty, I think that's more in theory or even just a hypothesis, which without any real solid supporting evidence. So I guess we'll give Snake. I think the question was for Snake. He can have the last word.
Yeah, so we have an anti-fragile genome, which basically means that you can mess with it a little bit. Some mutations are worse than others, but we get along just fine with some instability, where we have certain thing like with cancer. There are certain things that are cancer causing, but we can get along with them for a while, and then cancer hits in old age. So some instability where ERVs can cause cancer, we can live with those mutations. And then there are some ERVs that cause instability in cancer-causing genes that interrupt those or combat them in some way by tagging them, or they can also they can upregulate or downregulate genes. And the fact that we see both is more consistent and actually exclusive to evolutionary mechanisms, because this is more consistent with the hit or miss type of method of evolution, because we're seeing from the same genetic elements, viral elements, we're seeing both beneficial and deleterious effects, upregulating, downregulating. Out of the thousands of ERVs we have, there are literally just a handful of like 1 or 2 that are absolutely necessary. So, and all of the rest of them are not. So that's way more consistent with the kind of shotgun approach of evolution, where most of these are going to hit in places that are really not going to do much. They're going to be basically neutral. We're going to see all their function all over the place, going up and down, beneficial, deleterious. That's not what we're going to see if we're looking at a perfectly designed genome. So, and then let's not forget that even in the best case, these are transposable elements that could not have possibly ended up in the same locations. Yeah. Is that it? Is that our final question?
We made it. 2.5 hours on just ERVs. I think that's pretty, Snake, this might be the most comprehensive debate on endogenous retroviruses that exists. And I think this is an important topic. And I, for one, want to do more of these types of debates where we focus on one line of argument, as compared to some of these debates where it's just a shotgun of arguments that we deal with. ERVs is an incredibly important topic, incredibly technical topic. And I'm glad that we had such a great audience on a very technical topic. So, of course, let's do a round 2 where we can go over some of the papers brought up, some of the arguments.
Yeah, like I would even say we go even more specific, like just talk about certain, like 1 or 2, a handful of papers at a time within a very highly specific topic. So, because at this point we've had the same, like, evolution versus creation debates like hundreds of times. So I think the audience is ready for
Amen.
a little deeper dive there.
Yeah.
Let's give them the meat and potatoes. So
yeah,
ERVs is to the evolutionist, the one of, some say the number one best evidence for evolution. So as a creationist, I think we should be tackling these types of debates and arguments, and I really enjoy this. I think this was a ton of fun. Snake, I'll send you maybe a few of the papers that I brought up. You can look through them and pick which one you want to discuss specifically, and we'll do some type of follow-up discussion on those. So again, Taylor, thanks so much. Brandon, great job moderating, brother. You're a superstar. You're the man with the plan. Very impressive. So
listen, next time you guys debate, it's going to be a rigid timer. I'm going to bring a blowhorn. And that's it.
We had a kind of a second debate there at the end.
Yeah, you did.
We did. Yeah, I'm not sure who asked that question, but kudos to you. Your one question was a ripple effect into a whole nother debate. Brandon, go ahead as host. Close her down with some closing words, thoughts, jokes. I don't want to put you on the spot or anything, but
man, I got to tell you, I actually learned a lot from that debate, not just from Donnie, but you as well, Taylor. I think you're, pretty well read on the topic, and not a lot of people are. It's one of those niche topics that's tough to get into, and kind of stay. I don't know what the word is I'm looking for. Like, to keep it interesting, people don't find that topic interesting like some of the other things do, but it is really important, like SFT mentioned a few times. So I appreciate the debate. I learned quite a bit from the both of you. I think a second debate's going to be excellent. I'd like to read some of the studies both of you have looked at. I think it's some pretty good talking points and something I'm definitely interested. So, I'm not going to run too long. I guess I'll just remind everybody again, head over on the LPP's channel. If you want to experience what it's like to stand on the surface of the sun in a blazing inferno, you can have that over there.
For me, I'm gonna get some food and get some beverages in me, and then I may just head on over there after. This was a fun, engaging, technical debate, so I'm definitely going to relax for a bit, and an important debate. So I appreciate the prep, obviously, that Taylor put into this, because to make for a nearly 3-hour debate on ERVs is pretty awesome, and I had a ton of fun. So those are my final words. Taylor, final words from you?
Yeah, it depends on what the ladies in my house are feeling, whether my dog and my girlfriend, if I go over the after show or not. But yeah, they're the boss for now.
I hear you. Okay.